Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Right. Welcome, everybody, back to In the Interim. I'm your host, Scott Berry, and I'm joined by Nick Berry today, Dr. Nick Berry, and we are going to do our second episode of lessons for drug developers from the world of sports. You can go back to our first time we did this, episode 14 of In the Interim, and we had a really nice episode on regression to the mean. Today, we're going to talk about the 10-run rule and futility. So maybe bear with us a little bit as we get to that. But I do want to revisit the regression to the mean and a prediction that Nick made. We did this episode last year. In May of last year, Aaron Judge was hitting 400. And for those of you who are not baseball fans, I last week had somebody from Iceland send me an email about the episode on the Panther trial. So we have people globally listening to this. A 400 success rate in baseball is a very, very high bar. Nobody's done it since 1941, and it's one of these mythical targets that somebody could be a 400 hitter. Well, Aaron Judge was batting 400. It's just his hits divided by at bats. In May, and that's early in the season, his rate was 400. We were talking about regression to the mean, so Nick predicted that by end of year, he would be hitting 330. Interestingly, Jim Albert, who we did another episode, I did an episode with Jim Albert about Bayesian statistics and sports statistics, his career in those, he guessed 320. And Aaron Judge's final batting average was 331. So Nick, you were off. By
one 1,000th of a, yeah. 0.1% off. But yeah, I think that was basically a weighted average of what he's done with some random semi-informed guess of his true batting average to get me to 330.
Yep. And by the way, it was an incredible season. 331 with the home runs he had was a phenomenal season.
We could do the same thing now. I think he's hitting like 225 this year, almost a month into the season. So the exact opposite. We can regress him to the mean in the other way this year.
That actually would have made for a good thing. Maybe we should figure out what he is, and you have to post another prediction. But let me talk about a different prediction you made, and this was a prediction we are coming off of. We are recording this episode coming off of the Masters golf tournament. So that was last weekend, and we're a big golf family. We enjoy golf. Even Nick's sister, Lindsay, who doesn't play golf at all, she loves to watch golf. So we were watching the Masters, and halfway through the Masters, 36 holes, 2 rounds, Rory McIlroy had a 6-shot lead. So he second place, and there were multiple golfers that were 6 shots back, but a 6-shot lead halfway through the tournament is a quite large lead. I believe it was the largest lead ever in the Masters at that. And of course, our family text goes to predictions, goes to statistics. And the question was from Nick's sister, Lindsay, what's the chance that Rory McIlroy wins the Masters? And multiple of us guessed, but Nick, you made a prediction of the chance that he win the Masters. And what was your prediction?
I think I said 70%. My math was, I think he probably has the best expected value of the whole field, just based on the fact that he won last year. He's playing really well this year. So I said he's probably the best player in the field this week. He has a 6-shot lead. And I figured that, his distribution was good enough that even though there was a lot of people with the opportunity to catch him, still a 70% chance of him winning.
And my prediction was 60%. And interestingly, Nick's mom prediction was 20%. It was quite different.
Which she knows golf, and she knows how variable it is. And that was the reason for her prediction. It was like, weird stuff happens. I just, it's one guy has to hold on to the lead. And so she guessed 20% because of all the variability and what could happen.
Yep. So at that point, to sort of come to this, interestingly, Scottie Scheffler, after 36 holes, was 12 shots back. And he's the number one ranked golfer in the world. Rory, I think, was the number 2 ranked golfer in the world in it. And Scottie Scheffler was 12 shots back. Essentially, very little chance he can win. Less than 1% chance he would win the golf tournament. How much less, but less than 1%. Interestingly, if you were 17 shots back, meaning after 36 holes, you were 5 over par. Rory was 12 under par. So if you were 5 over par or more, you had a 0% chance of winning because you were, quote unquote, cut from the tournament. Those players stop playing and they reduce, and the rule of the cut is the top 50 players, and I think it started with 91 players. The top 50 players and ties continue on into the weekend, the last 2 rounds. But they cut, and sure, I'll say the word futility, that these players no longer have an opportunity to win the golf tournament. If you were 17 shots back, happened to be in that tournament, where Scottie Scheffler was 12 back, but he got to keep playing. Now, what happened in the golf tournament, Nick?
So round 3, Rory struggled. I think shot 1 over par. So he went to 11 under, and the rest of the field played great. It seemed like almost everyone that was 6 or 7 shots behind him shot 5 under, 6 under, and he went into Sunday tied for the lead. So he blew his lead, right? It disappeared after day 3. Scottie Scheffler shot 7 under par and moved into contention. I think the number 1 player that was 12 shots back. And on Sunday, the whole field kind of struggled. Nobody shot 7 under. Nobody had a huge round. Rory eked out a win by 1 stroke. So he did technically retain his lead, even though he blew it and got it back at the end. Yeah. But the most important part is that I was the most right in our family group chat because I said 70%, which was the highest number. And it doesn't matter how he got there. There's no pictures on the scorecard. I picked the highest probability. Rory went on to win. So I claimed victory in the family group chat.
An interesting part of it was Scottie Scheffler ended up finishing 2nd place, 1 shot behind. And yes, he missed a birdie putt on 17 that eventually could have led him to a tie within it. So he finished 1 shot back. He had a very, very small chance of winning the golf tournament. In a different setting, we could have said, you're done playing, you have no chance to win, you're cut, you can't play in this setting. But he came back and had a legitimate chance of winning the tournament. And so I won't address your 70% being the right answer when we know more about the golf tournament, but you're technically right. If you evaluate the likelihood function of what happened, you had the highest likelihood function in that. But coming to this question about in a sports competition, do we stop the sports competition? And what does it mean? And you won't be surprised to see that we'll turn this into thinking about stopping clinical trials and what are the similarity of that. So Nick's brother Cooper, and if you're on video here, you can see Nick is wearing the team shirt for the Pomona Pitzer Sage Hens. Nick's brother Cooper plays on Pomona Pitzers baseball team. And in a recent game we were watching, he was playing up in Oregon, up in Portland, Oregon, and they were playing Lewis and Clark. And in game 3 of their series, they were tied at 1 each. The Sage Hens scored 6 runs in the 6th inning, and they went up 11 runs. They were up 11-0. And in the bottom of the inning, Lewis and Clark didn't score. And so after 6 innings, and it's a 9-inning game, so you're 2/3 of the way through the game, the Sage Hens were up 11 runs. Now, the game kept going. So the game did not stop at that point. In the 7th inning, they scored 1 more run to go up 12-0. Lewis and Clark, the Sea Otters, I think are their mascot, scored 0. And the game was 12-0. And by rule, the game ends at that point. It's futile. And there's a classic 10-run rule, and there's a 10-run rule after 7 innings that if one team is ahead by 10 runs or more, the game is over. So the game ended in 7 innings at that point, and we stopped. Now, we have futility rules in the Masters. We stop players who are not in the top 50. In youth sports, we do this quite a bit. We have a 10-run rule in baseball. It's a little bit different in sports with a clock. Baseball has no clock. It plays 9 innings, and that's kind of the clock. So games can go long in that setting. They do have travel, and they're dealing with that. In games like American football and in hockey and basketball, they do something called a running clock. So if a team goes up by 5 goals in a hockey game, they don't stop the clock between whistles. They let it run. They shorten the game. So this happens. Nick used to play select baseball, and they did something kind of interesting where after 3 innings, if you were up by 15, they might stop it. 4 innings, it was 10 or 8 or more. And after 5 innings, after 4, it was 10. And after 5, it was 8. So they had a graduated rule within those games, and the game is over. So why do we do this in sports, Nick? Why do we have these rules that stop the competition?
Well, I think the reasons behind them are confusing. There's a lot of, we'll call them stakeholders to this, but I think the implication of all the rules is that there's, I'll say, no point in continuing to play because the eventual, result is determined at that point. And I think in reality, it's probably, everyone understands it's just a sufficiently small probability of something other than the obvious thing happening. So we're willing to say, for the benefit of all involved, let's truncate the game at this place. We know where this is going. I think in youth sports, people got places to be. Parents got to get home. There's another game right after this. The tournament's running late. We only have the fields until 9:00. A lot of different reasons to go to it. For professional sports, or I'll call what Cooper does something where the school's paying for this and there's money. I mean, coaches have a stake in this. They're wasting resources. If you're pitching guys late in the game that you don't want to throw, or silly things happen like, oh, we lost, we have to play 2 more innings. Let's put an outfielder into pitch because I don't want to waste a pitcher. And things start to get wacky in that regard. And so maybe we should just call the game at this point. But I think it all boils down to everyone knows what's going to happen. What's the point in continuing to play, sort of, is the idea behind all of them.
Yeah, there's also a concern in youth sports that if one team has such a lead, it becomes almost unsportsmanlike for that team to be trying hard to win. Are they going to win by absurd amounts, for example? potential injuries. There's aspect. We don't have futility rules in sports. Now, we do have some, and I'll come to those. But largely in an American baseball game, if one team is ahead by 25 runs after 8 innings or 7 innings, they keep going. In those sports, they don't stop them. Now, people have paid to go watch that game. They're selling beer, though they don't sell beer after the 7th inning, but they're selling beer at an NFL game, at an NBA game. A team could be ahead by 40 points in an NBA game, and they keep going in those games. So pro sports are a little bit different. They do worry about injuries, but there are no futility rules in pro sports, which is somewhat interesting.
Well, you mentioned the fans. The fans do have futility rules, and this is if you're at a baseball game and a team's up 20 runs, the stand's empty. People, to them, I think it's more important to be in the car before traffic hits than it is to watch your team get smothered by 25 runs. I'm definitely a stick it out to the end. I paid to be there. I went through the effort of getting there. I'm going to watch the 9th inning of this game, but the stadium does empty in those cases. So fans have their own, futility rule that they've created in their mind for when it's over.
So can we make a mistake with a futility rule? Could we have stopped that game that Cooper's team played up in Oregon? They were ahead 12-0 after 7 innings. They could have lost that game if they kept playing. The rules are a 9-inning game, and the Sea Otters could have scored 13 runs in the 9th inning and won that game. It's possible in those circumstances. Just like it's possible that we could have cut Scotty Scheffler from that golf tournament and said, you don't have a chance to win. You no longer get to play. And he could have won that tournament by the end. He didn't. But in another setting, we have stopped competitions that would have flipped and the other team would have won.
Yeah, for sure. I think there's a heuristic. This is Bill James, the famous analytics, the father of analytics. I think people give him credit for that. He has this, I called it a heuristic. I think it's a good word for it, for college basketball games about when he thinks the game is over. And it's a simple calculation. Like you take how much you're winning by, -3, add a little bit if you have the ball, and then I don't even know what you do from there. Something like, yeah. So there's some weird little algebra scheme you do, and it says this game is over, or this game is not over. And I bring this up partially because I wanted to talk about this example, but when I was in college, Texas A&M was playing in the NCAA tournament. We just finished March Madness as well. And Texas A&M was playing Northern Iowa in, I think, the round of 32. And they lost. They were futile according to Bill James' algorithm. We were down 12 points with 42 seconds to go or something like that. And doing the calculation, that's impossible to come back from. And I have 2 good friends that were at the game. They left. Yep. They left. It's obvious it's over. Got to beat traffic. They're in the concourse walking away. And my alma mater came back and won. Alex Caruso, of now 2-time NBA championship fame, was on that team. And it was, the comeback of a lifetime for me in school. And so it actually happened. This is futile. This would have stopped for futility according to a popular rule, and it switched. Weird things happened. The game reversed, and Texas A&M won.
So there are some cases where you can't come back, but in all those situations, it's possible. The probability may be very, very small, and we'll get to that question of the probability of winning to that. There are other cases that we do have futility rules. In pro sports, when you're playing a 7-game series and one of the team goes up 4-1, they've won 4 of the first 5, they stop playing. they don't play the 6th and 7th games. And in that situation, it's impossible for the other team to win, so they stop playing. So they don't say, well, people have bought tickets, we're going to sell food, we're going to play games 6 and 7. They do stop playing and they go on. So there are some binding's the wrong word, but there are cases where it's impossible to come back and they stop playing. Another example of that is golf, where when you do match play, like in the Ryder Cup, you play who wins the most holes on 18 holes. If you're up 4 with 3 holes to play, the golfers stop playing. They don't play the last 3 holes. The match is over.
You're mathematically eliminated. And that's...
You're mathematically eliminated. Yep.
It's an obvious way to do futility, right? Like, you don't need a model or any complex reasoning to understand why that happens, or to determine if it's a good rule.
Right? Yep. So let's come to questions of how we might suppose a professional league came to you and said, we want to institute stopping rules that when the game, reaches a particular point. Bill James had a rule. We should stop it for safety, for sportsmanship. How would you do it?
Carefully. I would start with a bunch of data. I think step one is get every single game that's ever been played at that professional level, and specifically with data about probably just the score over time. I think one thing you can't do in this is put, and we'll come to this later, but you can't put any preconceived notion about the quality of the teams into this calculation, right? I think it has to be a hard rule based on some just like, observable score difference or something like that. So yeah, you start by plotting all of these score differences over time, overlay them on a plot. And then I would probably choose a reasonable time in the game, maybe 3/4 of the way through. You don't want to stop anything, while there's still an immense amount of variability in the game. So I may start 2/3 or 3/4 of the way through, or some number, and I draw a cut there on that graph that I was making and say, okay, what point on the y-axis, what score differential can I choose that first no one has ever come back from? I'd probably start there and say, okay, if you're up 37 points with 10 minutes to play in a basketball game, the game is over. I think that is a useful starting place. It's probably not the rule I would end up with, but I would start there. And then maybe I say, okay, what about 1%? I say, okay, what point on this line, do you have a 1% chance of coming back from? And that's probably too big of a number. In baseball, every team plays 162 games. So multiply that by, 15 to get the total number of games played a year. That might not be the right math, but 1% of those games is still a lot. You don't want to have that many flip-flops of things that you stop. So I'd probably choose a number like 0.1% or something like that, or 0.05% of games, and draw a line there. You could do this with a curve as well. Maybe I do 2/3, 3/4 of the way through, 5/6 of the way through, and 7/8 of the way through, or something like that, and draw that same point at those 3 lines, and you could stop for futility at any of those points.
So you're getting at something. You're interested in this, at looking at different points in the competition, where what is the chance the team that's trailing can win the game? And you mentioned when that's below 1% or 0.1%, that the team that's trailing could come back and win, that this is a candidate game for stopping, that we've reached it. Getting to this point of predictive probabilities. So you're building this model, and you talked about getting all of this historical data we have on competitions, whatever sport it is, that you would build that. What is the probability this team can win at this from this state in the game? We have something like that in most of our sports, for example. And gambling is a huge part of sports within this, so you can get this. Our favorite baseball team, Nick and I are fans of the Minnesota Twins. They played a game yesterday. It was Wednesday, April 15th, and Minnesota was trailing the Red Sox throughout the game. They got behind, they actually were ahead 1-0, but they got behind 9-1 by the 6th inning. ESPN provides win probabilities, and you can graph this across the game. So they're doing this. So they're providing win probabilities. And it was not long by about the 7th inning in a 9-1 game that the probability that the Twins were going to win dropped below 1%. Now, interestingly, they scored 4 runs in the bottom of the 9th and they lost 9-5, but that probability never wavered from 99%. That game could have been a candidate. Maybe it doesn't reach the threshold. These are really natural things within games to look at. And then you balance, why are we stopping? What are the purpose to understand the risk? Because you could stop a game that the team would have come back and won, and that's a mistake to some extent. You've made a mistake by stopping it. You could also not stop a game that the trailing team lost. Now, it's a different kind of mistake where maybe the resources used in the remainder of that game were for naught because the game didn't change in that setting. We might consider that if we're trying to look at a rule for its ability to stop trials that are going to lose anyway. We would evaluate those criteria, those errors that we might make with a rule to stop sporting competitions, which of course have very different goals than clinical trials, which of course we're going to get to. I also have a friend. Yeah, go ahead.
Sorry, that quantity that you keep talking about, you've referenced both probability of winning, but also predictive probability. I think it's interesting. The first podcast we did about regression to the mean, I think we kept telling people, at least non-statisticians, that's a hard thing to get your mind around, right? You see data and you're like, okay, that's what I'm going to believe going forward. So we were trying to tell people, you need to regress this towards the population average, even though it feels weird to do. I think the predictive probability is the exact opposite. Everyone that is making decisions about these games intuitively understands what you're talking about. They understand like the variability left in the game, and they know why a 4-run lead in the 4th inning is different than a 4-run lead in the 8th inning. And they don't need to describe it as information fractions and variability and things like that to make that point. But I like this topic because it's so intuitive, and it's just like the root of decision making in general is this like predictive framework that we're working under.
Yep. So you brought up this idea, and this is going to be in sports. It seems like such a natural thing. And by the way, that's the goal of these episodes, that you take something in sports that seems so natural and understand, and then you flip it to clinical trials, which, that's sort of how I think. And it's not uncommon in a clinical trial scenario that I explain something like a sporting competition, but trailing by 5 early in the game means something very different than trailing by 5 late in the game, because you have less time to have a large differential to reverse that deficit that you have in the scenario. It's such a natural thing in sports, where in clinical trials, do we build futility rules off of the observed effect? How do we do that? The win probability in sports incorporates that. So in that game where the Red Sox were playing the Twins, the Red Sox were up by 5 runs after 5 innings. The win probability for them under that scenario after the 5th inning is about 90%. When the game goes to the 6th inning and the score doesn't change, it's still 5, the probability the Red Sox win goes up because there's less time left to flip that. So the same observed effect at different times have quite different predictive probabilities. It's a very natural thing in sports to understand that part in the win probability. Okay, so let's flip this to clinical trials. And we've already set this up a little bit. So what does this have to do with clinical trials? Many clinical trials are set up much like a sporting competition, especially phase 3 trials. Earlier stage trials can have goals that are largely learning, estimation. Those may be a little bit harder. You should have futility rules in those settings, but let's think about phase 3 trials because they're the largest, the most expensive, but they have very well-defined rules of quote unquote winning that trial. They're targeted on demonstrating one treatment is better than another treatment. In a sporting contest, it's almost like a player has to play, the best player in the world to show they're better. They have to show that they can win that game. We have very well-defined rules in sports about who wins the game. Now, in clinical trials, we also have really well-defined rules that we're trying to see that this treatment is statistically significantly greater than another treatment. So again, thinking about this question, like before we talk about how to do this, I asked you the question about why we might stop a sporting competition. Why might we stop a clinical trial before the end for futility?
Again, I think there's a lot of reasons. There are patient ethical responsibilities of the trial, and if it's looking like the treatment has no chance of being successful in a clinical trial, is it ethical to be randomizing these patients, to be experimenting on them? For a sponsor, there's certainly monetary constraints. Some of these drugs are extremely expensive to administer. It's extremely expensive to follow subjects up. We often can roll over into some open label extension where a drug that might be better for these subjects can be administered to them after a trial stops for futility. I think there's a ton of different reasons. Patient safety and cost are probably two of the most prevalent.
Yeah. So there's strong reason that if you're running a trial and it's very, very unlikely the trial is going to be successful, there are benefits of stopping that trial. And the ethical parts of it are huge. You're asking a patient to contribute to science, and if you look at the data, they're no longer contributing to science. The answer's largely been, the question's been answered at that point. Now, you can, any futility rule in that trial, that, by the way, there are some strange trials where mathematically the trial cannot be successful, but those are very, very rare, where it's integer valued outcomes and no number of events can flip the outcome. Those are different type trials. Most of these trials, you're now addressing similar type questions. And so we are, as clinical trial designers, asked to create futility rules. So the NHL is not asking you this, or Major League Baseball, but how do we create futility rules for a clinical trial?
Yeah. Let's see. So I think it's largely the same as what I described for like the NHL rule, with probably one major difference is that I wouldn't start by collecting a ton of data. I don't think I'm going to start with all the clinical trials that have ever been run and looking at their curves. I know the data generating process, or at least I have assumptions about the data generating process and what I think is likely. So I would probably start by you could think of it as simulating 1 million different paths through this. And maybe I think of this at the beginning as I simulate every patient, or I look really often, I take benchmarks throughout this trial, maybe 100 benchmarks. And at each of those points, I can maybe I fit a t-test. Maybe I, do fit some simple test for now to say how much better is the active arm doing, how much worse is the active arm doing. And then I go through the same exact now I have those spaghetti plots exactly like I had for sports. I had the score differential over time. I have treatment effect over time. I would probably start again. Maybe I don't want to start until halfway through the trial. I would say, okay, at what point is it impossible? Did none of my 1 million simulations come back to success? Where the treatment was so bad that it couldn't get back to being successful. And again, like I said with the other example, that's not necessarily, it's not a good futility rule, right? Something that never allows a reversal is probably a little too conservative for my liking. But that probability of reversing is really important. So I again want to limit it to maybe something like 1%, or in sports, I use 0.1% because we're doing this over and over and over and over and over. And I didn't want a bunch of these, but in clinical trials, I think a predictive probability of 1% is still fairly conservative for a futility rule, but something in the 1 to 5% range is probably where I would start in trying to cut that probability of reversing after. And again, I'm looking at trials that I simulated from start to finish. So in reality, I know when I stop one of those simulated trials for futility, what would have happened. And I think that is a really important aspect of the simulations, that I get this counterfactual look at all of the simulations and can do that. And so again, I'm drawing a spaghetti plot. I'm placing benchmarks at maybe half, 3/4 away through the trial and giving myself futility rules at those points. So the same exact process with the data generating mechanism being different.
So there's this same quantity that was this win probability that you can go to ESPN.com and look at. We build that in clinical trials, and you can build that through statistical modeling on what is the outcome. Is an event outcome? Is it a change from baseline outcome? Is it a responder outcome? You can take that outcome and build a mechanism for calculating what's the probability that the treatment is going to demonstrate benefit by the end of the trial. What is its win probability? That's something in clinical trials called a predictive probability. You may have heard of a conditional probability. Conditional probability is similar. It's a little bit different in that a conditional probability, you assume you know the truth about the team or what the treatment is, and then you calculate that. A predictive probability estimates it from the data at that point with its uncertainty in calculating the probability of winning the trial going forward. So there are different mechanisms. Bayesian tends to be predictive probability, where conditional power is a pretty simple taking the observed rate that you see right now, usually underrepresents variability. So I don't love the number that comes out for conditional probability, which is why we use Bayesian predictive probabilities. And then we look at that for its potential stopping the trial. When that probability gets low, you can look at stopping the clinical trial.
To keep our link to sports here, you're talking about conditional power or conditional probability of success versus predictive power, predictive probability of success. The calculation done in sports is different than the calculation done in a clinical trial, because in sports, we know a lot about the two teams going in, and we leverage that information in a way that's not common in phase 3 clinical trial, as Lacey were saying. So if I was doing a predictive probability in sports and a team's winning 9-1, like the Twins game you were describing, I am going to shrink my posterior probability of, the quality of those teams massively towards it being more of a 50-50 game going forward. something that conditional power does naturally. If you were to say, I'm going to calculate the conditional probability of the twins winning, given that the teams are equal, it's something you could do with a conditional power and get that number out. In a predictive probability sense, you would just have a prior probability that the teams are pretty close together, because they're professional sports teams. They are pretty close together. And even though you observe 9-1, doesn't really change what you think about the team as a whole. It's just that the game is random. So your predictive probability in that sense is actually probably pretty close to a conditional probability of success at that same number. And so the huge amount of prior information, I think, kind of changes the decision-making of that predictive probability.
But that can be incorporated in clinical trials, where you might even want to build a stopping rule that says we want to stop when an optimist about the drug thinks the probability of winning is below 1% or 5% kind of thing. And you use a Bayesian prior that gives a reasonable probability, an optimist estimate of the drug's effect. And even when that optimist thinks the trial is unlikely to be successful, you stop. By the way, it alludes to this in the Bayesian draft guidance, that you can use this design prior sort of thing. You can do this for the futility. So it's something that can be incorporated in clinical trials for sure.
Yeah. Yep. That makes total sense.
Now,
that's a Jay Cadane idea.
Jay Cadane's idea actually is a really nice idea, that you take multiple clinicians with varying views across the spectrum, and when all of those clinicians are convinced of it, then you stop. So you might need to go longer to demonstrate that you think the drug is effective to a pessimist, and likewise to stop when you think it's not effective as well. It's a really neat idea, actually. Now, this characteristic, we spend a great deal of time simulating different rules, and we calculate those error rates that we talked about before. So you can make a mistake with a futility rule. You could have a futility rule that stops a trial that would have gone on and flipped and reversed. And you say there's very little chance for the treatment to win. And we calculate that's 1%, for example. We've done trials where the predictive probability of success of that trial is 2%, and it stops for futility. It has a 5% futility rule. The trial stops. By interpretation, that there's a 2% chance that trial could have come back and been successful under that scenario. It's just deemed that, taking into account the pluses and minuses of this, that it's a good thing to stop that trial. Now, that decision is made ahead of time in those circumstances, and we simulate multiple rules. We look at error rates. We also look at what happens to power. When you have a futility rule for a treatment, you may decrease its chance of success, because some of those trials, those spaghetti plots that Nick talked about, they hit the futility rule and they would have reversed and gone on to win. You reduce power by that. If you have a very, very aggressive futility rule, an example of this would be halfway through the trial, if the predictive probability of the treatment winning is less than 50%, we're going to stop the trial. That would be a strikingly aggressive futility rule. Essentially, it means that the treatment is observing something very close to what it needs to win by the end, which gives it kind of 50-50. If it stays there, it wins. If not, it loses. And you stop the trial there, you're going to reduce power by very large amounts. Nick and I are looking at an example when that probability can drop power by 25% in your effect size, because you're being so aggressive at stopping it and you don't give the treatment that chance to go on and be successful. A 20% rule, by the way, and I'm going to say 20% because there's an interesting story, futility story to this. In the example we're looking at, can have somewhere a 5 to 10% reduction in power. 20%, if you think about a sports competition where the team only has a 20% chance of winning. That happens a lot. Those are exciting games, by the way, where a team scores 2 touchdowns very close to the end to win. Golfer comes back from 5 shots back on the last 9 holes. That's not even 20%. Coming back by 3 shots is sort of 20%. That's a common occurrence. Well, that rule is used in futility for futility in trials. And there's a famous example when that rule was used. And it's a famous example where people think futility might not be a good thing in clinical trials. And we'll kind of get to that. It's the Biogen example of aducanumab. It was an Alzheimer's treatment. And this was, 5, 6, 7 years ago. Alzheimer's treatment, a disease-modifying therapy. They're running 2 phase 3 trials. And they did a futility analysis where they looked at both trials, a predefined futility analysis. And they looked at both trials and they stopped both trials for futility. Interestingly, they stopped those trials, but they got follow-up data on one of the trials turned out to be statistically significant. In Alzheimer's, that's a huge deal. The other trial was not. And there was a lot of discussion about this was a bad futility rule, these types of things. Their rule for futility was that if either of the 2 trials had less than a 20% chance of success, they would stop both. And if you think about that in the sports contest, that's a really, really aggressive futility rule. Now, it was a bit more complicated because one of the trials, the conditional probability of the one that won was 60% when they stopped. So it was a controversial application of futility rules in the scenario. I mean, to some extent it was right. The other trial was not successful. But in hindsight, I think Biogen would have done it differently, and they were not necessarily pleased with the futility rule in that scenario. Now, I did a debate with Paul Eisen from USC on whether we should be doing futility rules in Alzheimer's trials. Now, you can go get that and you can find that debate. And I did win the debate. 80% to 20% people agreed that we should be doing futility rules in Alzheimer's trials. I feel like if I would have lost that, I feel like it's such an yes, we should absolutely be doing that. We should be doing futility rules in every phase 3 trial. That if I'd have lost that, that would have been like Rory losing, leading by 6 after 36 holes sort of scenario.
The aducanumab example is interesting because earlier while we were talking, I was thinking about how the value you choose for your futility rule almost creates this sort of implied utility on what you're valuing in your clinical trial. If you have an aggressive futility rule, you're probably saying it's really expensive to continue. I only want to spend this extra money if it's likely that we can win. So the implied utility of that aducanumab futility rule is something like we need 2 successful phase 3 trials or bust, is sort of the implication of that. And that was probably what was going through their mind while they're constructing the rule. Like in order to get this approved, we need 2 phase 3 trials to meet this threshold and the FDA will approve us. And, clearly that's not what happened at the end of the day, but you can see how they got there a little bit. And I think the utility that comes out of this is out of your futility rule is interesting. And, you say a lot by not actually saying anything on purpose.
Yeah, no, we spend a great deal of time building these predictive probabilities. In that scenario where you have 2 phase 3 trials running at exactly the same time, the idea that you do conditional power only in that trial and you ignore the data from the other trial, the fact that the other trial had a 60% chance of winning meant that it had a good effect size. If you would have done regression to the mean and shrunk over the 2 trials, you probably would have got a much higher probability for the second trial of being successful. And then maybe that would have eventually been successful had they run it out to the end. So a huge part of what we do as statistical consultants is build good predictive probabilities so that we're making good decisions. In almost every clinical trial, like Alzheimer's, like stroke, oncology, you have information on patients that's short of the primary endpoint. They're not all as simple as success and failure, and you know everything about everything in the trial. You have incomplete information on this. So we're building predictive probabilities that are using maybe dose response modeling. They're using longitudinal modeling of early clinical outcomes to later clinical outcomes based on their predictability. We spent a ton of time getting those predictive probabilities to be really good so that they make really good decisions in clinical trials. And I suspect that was not optimally done in the Biogen example. Okay. I do want to make reference to a nice paper by our colleagues Roger Lewis and Barbara Wendelberger in JAMA. There's a futility in clinical trials. There's a JAMA series for methodology in clinical trials, and they write about futility. You can read more about those. But unfortunately, Nick, I think we've hit the futility for this episode.
Yeah.
And that's more time and resources based here.
This is
the fact
a time-based
that we're losing.
sport. Yeah, right.
Yeah, yes.
Clock ran out. We didn't lose. Clock ran out.
Yes. Yes. So we appreciate you all joining Nick and me in this deeper dive into the intersection of sports, statistics, and the science of clinical trials. Until the next time, we'll be here in the interim.